And welcome back. We are going to jump into the November 2024, but we're going to just deal with the biological terms. As we go through these terms, we'll also be discussing other terms that could be similar or easily confused also for the same one. So the first one is a nucleic acid that carries hereditary information. All right. So obviously we have, when we speak of nucleic acids, I just want to kind of get back into black. When we speak of nucleic acids, we have our DNA as well as our RNA. So then now the question is, then what do they do? The DNA carries the information, but the RNA moves the information. So if we think of the information found within the nucleus, when that DNA information leaves the nucleus through our pores, so it leaves our nucleus into our cytoplasm, it moves using RNA. So when information is moved, or if a nucleic acid is moved or that information is moved, it will be RNA. But because our information is just carried, it would then have been DNA. All right. We are then moving into the next one. So the next question, the next biological term would be chromosome condition of a cell that has a single set of chromosomes. So when we speak of chromosomes, we know that we have haploid and we have diploid. All right, jump into understand meiosis if it doesn't make sense and where it would be. But basically, if we're speaking of a single chromosome, we'll be speaking of a haploid. And then if we obviously talking about our two, we'll talk about a diploid. So if you go into meiosis or even into the male and female reproductive system, understanding how the egg is prepared and how the sperm is prepared, that will give you a clear indication of when we would have an end product haploid or an end product diploid. So this is basically meiosis 1 compared to meiosis 2. All right. The next biological term that we're speaking of, the structure that holds two chromatids of a chromosome together. So what I always love to say is that, sometimes when you read a question, obviously you're stressed, you have these 3 hours, you really need to kill it. And you can so easily forget such small words because we out here cramming the eye and all the parts of the eye. So break up the question. So we have two chromatids. So we know we're speaking about something about a chromosome, right? And we know our chromosome normally looks something like that when we draw it. So again, I have no idea what I'm talking about. So I'm just reading. So let's say the structure that holds two chromatids of a chromosome together. So you see, I didn't understand what was happening, but then I'm reminding myself that a chromosome looks like this. And I know a chromosome normally has these two pairs because when I'm speaking of meiosis, something must break this thing and then it becomes two pairs and then it gets pulled to the end of the cell and blah, 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 blah. So all I'm trying to say is that biology, try to really have a good grasp on understanding more than a biological term. So that's why I'm running through these questions, but more for, let's think of it in the big picture, because I think these questions are so isolated and really they don't even carry the amounts of knowledge that you could possibly have, but it's 9 marks you cannot risk. So obviously we understand that it's something in the middle. So in the center is how you could remember it. Oh, sorry, I'm actually writing with an eraser. It's something in the center. This thing is in the center, so that's how you remember it. And obviously we all keep talking about these chromo. So it's a centromere, as if we're saying a chromosome extra. So that would then just be a centromere. All right, so let's move on to the next question. I believe it's number 4. Number 4, the type of RNA that carries specific amino acids to the site of protein synthesis. So again, in the question, I'm like, oh, I don't know. So type of RNA. So I would literally write, what are the two types of RNA? mRNA as well as tRNA, right? Again, now it talks about carry specific amino acids, but I remember when I was doing translation transcription, I couldn't even tell the difference. Like those two T's were confusing me, but I need to get this 1 mark. So let's find the answer in the question. To the site of protein synthesis. So we understand that this one is going to the site. So it's a transporter, right? And it's going to the sign of amino acids. Amino acids. Remembering that the amino acids is in the cytoplasm, but the mRNA works in the nucleus, because this has to take a nucleus. Something there is funny. Anyway, the mRNA is in the nucleus, and the tRNA is actually like the transporter with our codons outside in the cytoplasm around the amino acids. So that's what I would use. So actually, sometimes it does help to just scribble out everything you have in your head and read it out to yourself. So yeah, the answer there would be tRNA. So I'm not even telling you guys the answers. I'm just helping you guys understand how to get it right always. Okay. The analysis of DNA samples to identify individuals or relationships between individuals. I have a good idea that in my textbook, this thing had things like this. I don't know if you guys also thought of the same thing when I read that question. I'm just like, what DNA? Because I know it's not going to be in any of the questions that I've been cramming before this exam, but let's use it. So let's go. The analysis of DNA samples. So I know I'm talking about DNA. Let's get that down again. To identify individuals or relationships between individuals. So this is kind of nice because this terminology explains what it says. So if you profile someone, you put them in a certain category. So when they ask you, like, what is your, like, Instagram profile? What's your modeling profile, for an example? It puts you as an individual in a certain type of modeling gigs. You know, if you are going to be doing natural hair, if you are going to be doing maybe like the modeling types, if your body is like that. So it's in the word that the fact that they're categorizing you as a certain relations for certain individuals is the fact that they're putting you in a certain profile. So this would be DNA profiling. You know me and my spelling, y'all, as long as y'all get it right. So yeah, so that one actually, so let's see the question, how we also could have gotten it wrong. So at my first read, obviously the analysis of DNA, I actually was already thinking of genotypes, because you can imagine I have all this information in my head. But then it helped me by saying samples. So then it reminded me of that all those black strips. And those black strips need to carry something. But I think one of the biggest things as I read the statement is that section is often overlooked, like DNA profiling, because everyone's like, yeah, but I'll get it, but I don't understand mRNA and tRNA. And we spend so much time understanding like translation and transcription, and such a small biological term could slip in the cracks. So that's why it is quite important. Understand your terms in relation to what they're talking about. Try not to cram. All right, the next one, a representation of the number and structure of all the chromosomes that occur in the nucleus of a somatic cell. So this one is a biological term that we'll kind of have to know. So this one's just a karyotype. So if someone maybe doesn't remember, when we were speaking of different mutations or things that could go wrong in our cell, we draw it out. Oh, I keep writing with my razor. We draw it out like something like that. And then like the last one is like a double. So that would be our karyotype. So that's what we're talking about. We're talking about the structure of all the chromosomes put together. And obviously that last chromosome, I just drew it like that because that's the one that would be telling us our mutation most of the time. All right, I'm an amazing artist, guys. Imagine. Anyway, the next one, a group of similar individuals. So I like these ones. So a group of AA that are able to interbreed to produce fertile offspring. Okay, I went ahead of myself. It wasn't the statement I thought. So a group of similar organisms are able to interbreed to produce fertile offspring. So that means this must be the same thing that gives you children. So this one's a funny one, actually, in essence. You know, we can't just give you the answers. We really have to help you understand how you would have seen it by yourself. I'm trying to think of a really beautiful. So maybe jump down into the comment section. Let me know. The answer is species. But I'm just trying to help you. If you were to just look at the wording, what in the wording would have told you that? So as soon as I said a group of similar organisms, I was already thinking of genetics. So I was now about to cross an A, B, B, C, C. Then it continues to say to interbreed. So again, I was thinking I'm going to cross, like I'm going to like create an end product or something. Then it says to produce fertile offspring. So obviously now I'm thinking of the fact that a chicken has to be with a chicken to make another chicken. A cow needs to be with a cow to make another cow. And basically what we'll be talking about is that the similar species can interbreed to produce fertile offspring. So like a cow can't be with a chicken, for an example, to give you fertile offspring. So yeah, so just in the statements, we're really trying to see if we've done minimal studying, how we could have used the statement itself to ensure we get our answers right. All right. The next is the phase of meiosis. So again, we're going meiosis. So where paired chromosomes are arranged at the equator. So this one's kind of nice. If you go through all the stages of meiosis, we want one where all our chromosomes, sorry, that's my equator, are lined up at the equator, right? Now, there are two meioses. So you must remember that it's either we have a chromosome at the equator, or, I can't mess it up, in meiosis 2, it wouldn't be a full chromosome because they would have split at the end of meiosis 1. So then this is then what we would have, because we wouldn't have a full chromosome lined up. So to have a chromatid. So basically, because it's a full chromosome, we know that it would be meiosis 1. Then in meiosis 1, we still have a few phases, right? So we know just after this, it gets pulled to the 2 poles. But when it's in the equator, it's a M because it's in the middle. So that would be metaphase. All right. So basically, we are saying we are in metaphase 1. So it is kind of important, if you want to get all your marks, that it's not just saying it's meiosis 1. Again, let's see, a phase of meiosis. So again, it did use the word phase, so that at least helps us that we want to be specific, which not just meiosis, but the exact phase within our meioses. All right, the last one. The biotechnological process that produces genetically identical organisms. So I just saw biotechnical and I said in my head, I can't even pronounce that word. Can I use the rest of the sentence to get my mark? So let's go. It's a process. It's a process that gives us genetically identical organisms. So let's draw it out because it's something that will give me 2 things that look the same. Okay. So obviously we know DNA replication does that, but that is again not a process. It's like it's within, it's inside, right? Obviously, it's that big word that gives us the biotechnological. So that word also tells you that it's a technical process, something man-made, right? Also produces genetically something man-made that can redo DNA information, right? Making it exactly the same. So then obviously it would be cloning. All right. So again, in the word, biotech would actually tell you the process, but you need to figure out where else can I get identical organisms. So again, I know I was also thinking of the female reproductive system, where where we're trying to like go through osteogenesis and spermogenesis could also look something similar, but again, it shows you that it's not sexual. It says organism. So this is an organ that we're trying to do. It has genetic information, so understanding that DNA replication could also give me is also a good guess. However, I think the big giveaway was the fact that it's technology, so it is man-made, right? That big word that tongue-tied me there. All right, so I think a big takeaway is not just going through our biological terms, but reading the question and pulling out information that you do know, layering that to give you your 1 mark guaranteed. This is 9 marks guaranteed. Any question that I hope you will get correctly. Jump onto another quick biological test, and let's see if we can also get all 9 questions right, even with minimal studying.